Let's look at what happened in Wuhan that led to the worldwide lockdowns to see how virus detection and vaccines are developed today. People in Wuhan had always suffered from pneumonia due to the extreme air pollution in that city. So when 3 to 4 people developed atypical pneumonia, it was nothing new. Respiratory illnesses are caused by various factors, such as chemicals, air pollution, and toxic lifestyles. There is also a huge amount of scientific literature about drug-induced pneumonia. Over 600 known drugs can induce pneumonia, and this includes common drugs such as paracetamol, antibiotics, statins, and even aspirin. So, if the goal is to determine the cause of a person's illness, you would think that all possible causes would be investigated. But that is not what happened. They took lung fluid from a sick person, which would have all sorts of DNA and RNA genetic material in it, as well as various types of human cells and particles, many types of bacteria and fungi, and possibly contaminants also. They removed the cellular debris so that they were left with RNA and other molecules in the solution that probably came from around 100 different organisms. This is the paper where they did the first genome sequencing of the SARS-CoV-2 virus. In the method section, it states that lung fluid was taken from only one suspected case. And the important thing to note here is that they were not taking genetic material directly from a virus. Everything from now on is done in silico, which means in silicon, as in the computer environment. There isn't anything from the natural world. It's all a computer model or construct. Any large strands of RNA are discarded because the computer analysis and sequencing deals only with small strands. The huge number of small RNA molecules are sequenced on a computer. After sequencing, the computer further removes more molecules in a process called trimming to clean up the data. This left them with 56.5 million different pieces of RNA from who knows how many different organisms. So, how do we piece these 56.5 million pieces of RNA together? To understand next generation sequencing, think of a gene as a code of letters that specifies a specific molecule, and that molecule is usually a protein that has a specific function. The code that makes up the genes has 4 letters or 4 bases, and it's a combination of those 4 letters that goes into the sequence, also known as base pairs. C's are always paired with G's, and A's are always paired with T's, but the order of the very long sequences is unique to the organism being sequenced. All this information is stored in chromosomes, and the totality of all this information is called its genome. Because they never isolated a virus, they never took the genetic information out of the body of it, and so they couldn't sequence it from one long strand. Instead, they had 56.5 million short strands, from many organisms in the decaying tissue in the lung fluid. In order to match up the little bits by overlapping letters, they use two computer programs called Megahit and Trinity. And this is where the magic comes in that creates the fraud. Some of these little sequences match up. The end of one matches the end of the other, and you can put them together. They believe that by adding up the smaller pieces to make a bigger one, that they have the genetic material of the virus, even though the sequences that they are using for the alignment never came from a virus or any known organism. They construct something that is not really there in reality. From the millions of genomes created, they chose the longest and compared it to the bat SARS-CoV-1 virus from the outbreak in 2003, which itself was a theoretical genome constructed on a computer. They found it was 89.1% similar, but the SARS-CoV-1 genome was shorter than the config they had chosen. So, using the bat coronavirus as a template, they took things out, rearranged things, and filled in the gaps on their strand until it looked like the bat virus. So, without knowing if this genome is real, without knowing the origin of the original sequences, they declared they had the genome of their new virus. To prove the results of an experiment in science, you have to be able to replicate it. So what happened when others tried to replicate these experiments? They took fluid from a suspected coronavirus patient. They instructed the computer program to make the genome a certain size and look for certain sequences, and this helped to get them something close. But when they compared the sequence to the original SARS-CoV-2 sequence, they did not match. In proper science, not being able to replicate the experiment with the same results more than once invalidates the result of the initial experiment. But we are not talking about proper science here. We are talking big business. So they say they have found a variant or mutation of the virus, and this variant could be more dangerous than the original virus. It's the perfect fear-generating device to sell their product. The old way of doing all this, from discovery to vaccine development with tissue culture experiments, used to take around 10 years. But now, it only takes days to sequence a theoretical genome in silico. Then the digital file can be electronically sent by email to the various vaccine companies, who then can put the file in their synthesizer machines, which in turn, in a matter of hours or a day or two, make strands of RNA and DNA from the input. That gene sequence can then be put into a vaccine, which then can deliver genetic material to human cells and make the cells produce whatever product is coded in that genetic material. This allows companies to get their vaccines to market in no time at all. As for the PCR test for COVID, they didn't use a sequence for the COVID virus. They used a sequence from the SARS-CoV-1 virus. In conclusion, the in silico genome comes from a mixture of unknown sources. It cannot be reproduced without error or showing false mutations. It's a computer model of a theoretical genome of a theoretical organism that hasn't been proven to exist. So you can choose to remain blissfully ignorant, even if it means risking your health and life, being stressed, following the fear-mongering, and doing what you are told to do. Or you can know the truth and make informed choices. As for the blissfully ignorant, all is well, go back to bed.